The process was discovered in 1946 by two scientists, Joshua Lidberg and Edward Tatum, and they gave the clue that bacteria undergo conjugation process. Okay, so by this process, they are able to transfer the genes from one organism to another organism. This is called the horizontal gene transfer or the lateral gene transfer, right? Now, most of the plasmids which are carrying the genes for drug resistance are also carried by the same process, conjugation. And this is estimated uh, to be responsible for the most of the bacterial resistance in the bacterial population. So if in a culture you are having bacterial cells and some of them are F positive, they will transfer these plasmids between the cells in the culture, and all the population of bacteria will become drug resistant if it is having some kind of resistance over plasmid. Okay, so the which type of plasmid is most important for conjugation is called F plasmid. Though we have discussed this in our earlier classes that a bacteria can harbor at least or at a max up to 7 plasmids in a single cell. So E. coli cells have been reported that they can have up to 7 plasmids in a single bacterial cell. Okay, and how many types of plasmids are should be present in a bacterial cell? It is completely dependent upon the competent factor, the competence factor, which is the competency for a bacterial plasmid, right? So if the bacterial cell is competent for different plasmids, so different plasmid can coexist in the same bacterial cell. But if the bacterial cell is not competent for different type of plasmids, ultimately some of them will be lost, and that's how the number is maintained within the bacteria. Right, so actually this portion we have covered from T. Brown. So it is written somewhere here. So normally there are two type of plasmids. One is called as stringent and another is called as relaxed. Stringent plasmids are those which are generally present in low copy number, means generally having 1 to 2 copies per cell, whereas the relaxed plasmids are those which have higher copy number in a bacterial cell, as it is written here. So stringent plasmids are large because therefore they are present in low copy number in just 1 to 2 copies of plasmid, of the stringent plasmid in a bacterial cell, whereas relaxed plasmids are present in multiple copies up to 50 or more per cell, right? So first of all, they can be of two type, either low copy number plasmid or high copy number plasmid, called stringent and relaxed respectively. The second important thing is the compatibility. So it is written that several different kind of plasmid may be found in a single bacterial cell, including more than one different conjugative plasmid at any one time. So F plasmid is one kind of conjugative plasmid, right? So which is responsible for the formation of these pilus structures which are required for sexual conjugation in bacteria. In fact, cells of E. coli have been known to contain up to 7 different plasmid at a time. So they may have 7 different type of plasmids. So one thing is they can have 7 different kind of plasmids. The second thing is each plasmid is having some copy number, so which can be a stringent plasmid or a relaxed plasmid. So depending upon that, it may have 1 or 2 copies or up to 50 or more than 50 copies per bacterial cell, right? So how many different type of plasmids should be present in a bacterial cell? This is the compatibility factor of that particular bacterial cell, and therefore we can make different compatibility and incompatibility groups. Wait a minute, I'm having a query. I should be audible. At least my mic is working fine. Am I audible? Student, please confirm first. Okay. Trishla, having your network problem on your side, please fix it and rejoin again. Okay. Fine. So depending upon these compatibility groups or the compatibility of bacterial cell, different type of plasmids can be present in a single bacterial cell, including different kinds of or more than one kind of conjugative plasmid in a single bacterial cell. So one kind of conjugative plasmid is F plasmid, which we are going to discuss in this class. And depending upon this F plasmid, we can group these bacterial cells into two main types. One is called as, so in this text, when we are discussing this text, we have discussed episomes. So what is episome? I'm telling again. So this class is going to be a bit interesting because we are learning a few new terms. So episome is if a plasmid, as I told you that up to seven, for example, reported in E. coli cells, different plasmids in a single bacterial cell, and they are having different copy numbers. So you can suppose there is a large population of plasmids which can be present in a single bacterial cell inside the cytoplasm. They can be integrated into the bacterial chromosome, and once they are able to integrate into the bacterial chromosome, this integrated form is called as episome. I'm having a query again. I do not know what is the problem. Krishla, I'm confirming. Yeah. Okay, so this form is called episome. Now we are going to differentiate. If someone asks you what is the difference between an episome and an HFR, so now we are having two terms. One is known as so episome. We know any plasmid can integrate into the bacterial chromosome, and this integrated form is generally referred to as episome. One is called HFR. Sorry. So now HFR is having a full form, high frequency recombinants or recombination, right? So high frequency recombinants are those cells in which only F plasmid or the compatibility, that means this conjugative plasmid, is being integrated into the bacterial chromosome. So if a conjugative plasmid, for example, F plasmid, is integrated into the bacterial chromosome, then it is known as HFR, high frequency recombinants. If a normal plasmid, means any one, okay. So we are having 5 different type of plasmids, right? We have discussed all this in previous classes. So if you do not remember, here it is given. So we can classify or divide these plasmids in 5 basic classes. F plasmid, which is fertility plasmid or conjugative plasmids, right? Second is resistance plasmid, means R plasmids. Whole plasmids, degradative plasmids, and some virulence plasmid, for example, Ti plasmid of Agrobacterium tumefaciens. So all these plasmids, if integrated into the bacterial chromosome, then it is known as episome. Similarly, if a phage or bacteriophage, what are bacteriophages? Bacteriophages are the viruses. So it is also given in the same chapter. Bacteriophages or phages are commonly known as viruses that specially infect the bacteria. So these viruses or bacteriophages, if integrate their genome into the bacterial chromosome, then it is known as prophage. So now we have 3 terms: episome, HFR, and third is prophage. So episome is any plasmid. If it is integrated into the bacterial chromosome, it is known as episome, right? If an F plasmid is specially integrated into the bacterial chromosome, then it is referred to as HFR, high frequency recombinant. Why this is known so? I will tell you. We will discuss this in today's class only. This is known as HFR if the F plasmid is integrated. And third thing is if a bacteriophage, which is actually happening during the transduction process, so then bacteriophage will release the DNA into the cell cytoplasm and it will be integrated into the bacterial chromosome. Then the bacterial chromosome, which is having the phage or viral genome, is known as prophage. Or you can say the viral genome section of that particular chromosome is known as prophage, right? Now we have to discuss few more things. So I will be back over this portion. So there are two main type of bacteriophages. One is this we have discussed already. This is called lambda phage. So and the second one is called M13 phage. This is a filamentous phage. So you can see the structural differences between these two, but uh, they are better choices. They can be used as cloning vectors. Also, they can be used in gene sequencing experiments, and they are having some specific features due to which they are being used in cloning experiments, right? So we will discuss M13 in today's class. I will also tell you some specific features of lambda phage. So both of them, both the bacteriophages or viruses, though they have structural differences, but they have a common host. They can infect E. coli, and okay, so we will discuss, right? Before starting this discussion, let me finish conjugation first because I want to finish this portion in today's class. So the two scientists in 1946 discovered the process of conjugation, which require a direct cell-to-cell contact between two bacteria so that they can transfer their genes from one bacterial cell to another bacterial cell. And this is a kind of horizontal or lateral gene transfer, which is happening between two different organisms or two different related species. Showed that bacteria undergo conjugation, a process in which genetic information from one bacteria is transferred to and recombined with that of other bacteria. So not only they are able to transfer the genes or plasmids from one bacterial cell into the another, also they are able to recombine with their chromosomes so that they will change the phenotype of that particular bacterial cell, which is called recipient. So here also we will have two cells, one is donor, one is recipient cell. In conjugation, direct contact between the donor and recipient bacteria leads to the establishment of a cytoplasmic bridge between them, and this lead to the transfer of part or all of the donor genome to the recipient cell. It will take place. So actually the complete transfer of the bacterial genome from the donor to recipient actually require 100 minutes. But as I also told in previous classes that 100-minute time is generally, bacteria do not have a 100-minute time for continuous contact. And therefore, due to the environmental disturbances and environmental factors, this cytoplasmic bridge, which is called as pilus, is broken down, and therefore the gene transfer is hampered in between some way. So the donor ability is determined by the presence of these conjugative plasmids, and one is called F plasmid. It is the best studied plasmid. We know most about the F plasmid, right? So conjugative plasmid exists in both gram-positive and gram-negative bacteria. But again, most research on conjugation has been done in gram-negative bacteria, in E. coli. And we know E. coli is a gram-negative rod cell. Okay, so one student is having a question. Oh, sorry. So actually, Trishla is having some internet problem in today's class. Trishla, actually, we have discussed this, and I hope you were there in that particular class. I'm repeating this particular section because uh, there are few new students, and therefore I need to cover it again. Otherwise, I have given a glimpse of this. Okay. Right. So mostly two things here. One is this conjugative plasmids can be of different type, but we know most about F plasmid, right? Fertility plasmid. Why this is called F plasmid? F for fertility, because it is responsible for the transfer of genes from one bacterial cell into other by the conjugation process. Therefore, and this is a method of direct cell to cell transfer of genes. This is called fertility plasmid, right? And the second thing is this F plasmid, though it is present in both bacteria, gram positive as well as in gram negative, but most of the research actually is going on in gram negative cells. For example, in E. coli, we have studied E. coli mostly. So, based on this knowledge, what we have, we will discuss all the factors which are required for this transfer. So there are at least 25 genes, everything we will discuss. So these genes can be categorized into two groups. One is called MPF, mating pair formation group, and the second group is called as DTR group. So this is called DNA transfer and replication group. So the two groups, and there are at least 25 TRA genes or transfer genes, which are present in this F plasmid, in this fertility plasmid, right? So which help in this overall process of transfer. However, much less is known about the conjugation in gram-positive bacteria. For example, Bacillus subtilis and other gram-positive bacteria. You have many examples. So a recipient cell that has received the DNA as a result of conjugation is called transconjugant. I told you various terms in previous classes. So special term for the recipient cell which will receive the F plasmid from a positive cell, F positive cell. So this is called transconjugant. Right. Transfer of plasmid is generally intraspecific means intra mean in the same species. Inter means between the species. So intra term is being used and intra is referred to as in the same species. So generally the same species transfer of genes is with the help of this F plasmid, and uh, different cells of the same species, different bacterial cells of the same species. However, many plasmids have transfer this system that enable them to transfer gene between the unrelated species. Rarely it has happened, but actually this is also reported that with the help of this conjugation, bacteria can transfer the genes between unrelated species also. And this specific process, when it transfers the genes between unrelated species, so it is given a term called promiscuous plasmids, right? So these are responsible for transfer in the unrelated species. But mostly the transfer of plasmids via this conjugation process take place in the same population, means the same species. We have not discussed much, Trishla. You are joined at right time at least, and we have discussed this paragraph only, right? So you can read this out. Now the first type is called F positive, F negative conjugation, and we will discuss three of them. One is called HFR to F negative and one is called F dash to F negative. So first of all, the new students who have not attended the previous class. So this F positive are those donor cells, those bacterial cells which are having the F plasmid. F negative cells are simply those bacterial cells which do not have the F plasmid. Therefore, they are called F negative, right? So what will happen in a bacterial population, because there are so many bacterial cells, F plasmid, which is also called factor of the E. coli, is a prototype for conjugated plasmid in gram-negative bacteria. Strains of E. coli with an extra chromosomal F plasmid are called F positive, which function as donors. So the bacterial cells which are F positive and having an F plasmid are called donors, because they can donate these F plasmid to those cells which actually do not have these F plasmid, right? Similar is the case with you. If you are having more money, you can donate to some person which actually do not have money, right? Or some, okay. So similarly, if bacteria is having F positive, means they are having F plasmid, they can donate a copy to those bacterial cells which actually do not have them. Whereas the strains that lack the F plasmid are F negative, and they will behave like recipients, means they are very similar to those individuals which actually do not have money, right? So they do not have F plasmid, so they are called recipients. So they will accept the F plasmid from the F positive or donor cells. Conjugative function of the F plasmid are specified by cluster of at least 25 genes called transfer genes, as I told you, and they can be divided. Means what is their role? Their role is to determine the expression of these pilus-like structures which are required for the direct contact or to make a cytoplasmic bridge between two bacterial cells. Synthesis and transfer of DNA during mating. So it is also required for the transfer of genes or the DNA during this mating process via these sex pilus. Interference with the ability of F positive bacteria to serve as recipient. So second thing is which is not known. Okay, so that these transfer genes will interfere or disable the ability of these F positive bacteria, means those bacteria which are having these F plasmid, so they cannot serve as recipients. They are donors, right? Because when these genes which are present over F plasmid will express themselves, it will lead to the formation of pilus structure over the bacterial cell. So one function we will discuss here. So the pilus is responsible for this conjugal transfer of genes from one bacterial cell, that is called donor to recipient, right? So we will discuss here. And the second function we will discuss at the end of the class in M13, infection. So these are not only, so these pilus which are made by these transfer genes which are present over F plasmid are not only having role in the transfer of genes from one bacterial cell to another bacterial cell via these conjugation transfers, but also it is having some other role. So what is the second role of this? So here it is given. Right, so the second role of the pilus, this is the pilus, and this is the E. coli cell, right? So generally this pilus is a tube-like structure which grow over this, which is a surface appendage in this particular bacterial cell. M13 phage, which is called the virus of E. coli, it will infect the E. coli cells especially. It will infect or do infection via this tube only. So it will bind to the pilus-like structure over the bacterial cell, and this has been asked several times, this question, how this M13 infection will take place. So actually through the pilus only. This pilus is the attachment site for this particular bacteriophage, and through this it will release its genome into the bacterial cell, and then making different means so many number of copies. And most important about this M13 DNA, this is a single stranded DNA, but once it will inside the bacterial cytoplasm, it will make double stranded DNA, and the double stranded DNA of M13 is called replicative form because it can replicate itself, right? So we will discuss this in more detail if if the time permits or we have time at the end of the class, right? So we have discussed the two functions. One is in the conjugal transfer of genes, and the second function is in the infection process of M13 bacteriophage, right? Now, how this TRA genes are classified, so they can be divided into two groups. One is mating pair formation genes, yani those genes which are responsible for the formation of this pilus-like tube, which are responsible for the formation of these surface appendages, right? And the second group is called DTR genes, that is DNA transfer and replication function genes. So they are, so the functions are given. So first of all, MPF, mating pair formation gene, encode proteins responsible for the formation of these pilus and which are required for cell-to-cell contact and for the formation of type 4 secretion system. So sometimes this is also called type 4 secretion system, which is actually a channel or the cytoplasmic bridge through which the proteins and protein DNA complexes are being translocated from one bacterial cell donor to the some recipient cell, right? The second is DTR genes. So we know the function of MPF class. So MPF is having in the formation of this secretion system or the cytoplasmic bridge or this particular channel that is called type 4 secretion system or the sex pilus, right? The second is called DTR genes, that is DNA transfer and replication function. So they are encoding some enzymes, for example, relaxase enzyme. And this relaxase enzyme is specifically able to bind to this ORIT, which is present over the F plasmid. And it will create a nick at this ORIT. ORIT is origin of transfer, T for transfer. Means when this F plasmid is being transferred from the donor cell to the recipient cells, it will begin with this origin, ORIT. So actually, it may have two origin. One is origin of replication and second is called ORIT, that is origin of transfer. So relaxase enzyme will bind to this ORIT site, encode some protein that will bind to the plasmid DNA at origin of transfer, that is called ORIT, forming a complex that is called relaxosome complex. You need to remember. So first of all, we have discussed MPF, which will make the channel. The second group that is called DTR, DNA transfer and replication genes, which will make relaxosome complex, mean they will encode some of the proteins. These proteins will bind specifically to the origin of transfer. They will produce a nick within the ORIT and become transiently attached to the 5' end of the DNA single strand. So once the nick is being formed, they will bind to the 5' end of this particular DNA. They will remain bound until this will start transferring from the donor cell to the recipient cell, as we will see in one diagram. So one strand of the plasmid DNA is transferred in 5' to 3' direction, means the same direction in which the replication is actually happening from the donor to recipient. So the transfer is also in the same direction. The transferred strand is converted to the circular double-stranded form and then by replication, and both the X-conjugate bacteria are now will become F positive. So the bacterial cell which is actually recipient and do not having the these F plasmid, they will, after receiving this F plasmid, they will become F positive. So initially the transfer is from F positive to F negative. Once the transfer is over, this F negative will also become F positive. So there is one, so I also bring some of the questions also in today's class, gene transfer and recombination. So this has been asked in IISc. Okay, so if an F positive and F negative cross will take place, so what will happen? Obviously, this F negative cell will become an F positive cell, right? So this type of simple questions have been asked in different examinations, as you can find in these questions. So today we also do some of the MCQs, right? Therefore, this will spread. So this is a kind of infection, and it will spread by infection among genetically compatible populations, which are actually of the same species of bacteria. Now this diagram is depicting that how it is happening. So there are two cells, F positive cell, which is having an F plasmid, and second is F negative cell, which do not have this F plasmid, right? So what will happen with the help of these TRA genes and the two category of genes which are present over this F plasmid? One is called MPF, mating pair formation genes. So they will encode for the surface appendage. This is called pilus. Pilus is a product of the proteins which are encoded from this this TRA genes out of MPF, mating pair formation group. Then we have the DTR group, DNA transfer and replication. So with the help of DTR genes, so this is not shown in this particular, let me make something. So what will happen here, suppose it is having the origin of transfer or IT. Okay, so what will happen, the relaxase enzyme will come and will bind to this particular site. So suppose it is having some different color, and the relaxase enzyme will be able to bind to this particular origin of transfer. This is an enzyme E, the relaxase enzyme. It will produce a nick in this double-stranded DNA. So this nick, and it will remain transiently attached to the 5' end. We have studied this. Then this transfer will start through the cytoplasmic bridge from 5' to 3' direction, right? So this one strand will move from this donor cell into the recipient cell, and after this is transferred, it will finally being converted into double-stranded form. So after completion of DNA transfer and DNA replication, so it will undergo DNA replication, and this will also become double-stranded DNA or the plasmid. Now both the cells, F positive cell will remain F positive because it will again do the replication by rolling circle mechanism. Okay, and this will become double stranded. The F negative cell, which was recipient initially, it will also become F positive or the donor. Now it will donate this F plasmid, means both can donate the F plasmid to some other cells which are F negative, and therefore you will see complete populations of the bacteria which are F negative will by the spread of this kind of infection or the gene transfer, they will become F positive. All the population of the bacteria will become F positive in a culture. So this is very easy to understand, right? Now we will make it a little complex. We will discuss the second type, that is called HFR. So first of all, yes, right, I will share them, no problem. Actually, I have taken them from the MCQ. I will share the complete book as well. So the second type, let it make a little complex. Now I already told you what is HFR. HFR is, suppose this F plasmid is being integrated into the bacterial chromosome. Now this will become high frequency recombinants, right? So the bacteria is high frequency recombinants. So as we will see in the next diagram, this is bacterial chromosome. This is not single stranded. This is already double stranded. Only they have shown this. So here, if you see, they have shown the two circles. You will suppose you can think this is double-stranded, but here also this is double-stranded, right? So here, but they did not show the double-stranded form, right? So you need to suppose this is double-stranded. So what will happen? This F plasmid will integrate into the bacterial chromosome. Now this F positive cell, which was having this independent. So mostly the plasmid. So what is the definition of plasmid? Plasmid are extra chromosomal materials which are able to self-replicate themselves, right? So they are generally present outside this bacterial chromosome, which is the main nucleoid, the chromosomal region, genome of the bacteria. But suppose if this F plasmid or the conjugated plasmid is being integrated into the bacterial chromosome, now what can happen? Think of it. So now what will happen, because this F plasmid was having different kind of genes which are called tragenes or the transfer genes, which are of two categories. One is called MPF, mating pair formation genes, and second is called DTR, and DNA transfer and replication genes, right? So with the help of these genes are still present in the plasmid, but what will happen now, this plasmid, since it is being integrated into the bacterial chromosome, now it is called high frequency recombinant because it can, while this F plasmid will be transferring from the donor to the recipient cell. This is not the recipient cell. Okay, so when this will transfer from this donor cell to some F negative cell, so this is called HFR to F negative transfer. So at this in this way, it can transfer the complete bacterial chromosome along with the F plasmid, right? So you can suppose there are different type of genes which are present over this bacterial chromosome also, along with these stra genes which are present over the F plasmid. So all the genes or all the chromosomal material of the bacteria can be transferred from one bacterial cell into some another bacterial cell that is actually F negative or the recipient, and this is called HFR F negative conjugation. Now you are able to understand why these are called high frequency recombination strains, because they are able to transfer their whole chromosomal material from one bacterial cell to another bacterial cell, including the plasmid as well as the bacterial chromosome, right? So they are high frequency recombinations or recombinants. The F plasmid in E. coli can exist as an extra chromosomal genetic material, as we have seen in F positive cells. But if it is integrated into the bacterial chromosome, then it is called HFR. An E. coli strain with an integrated F plasmid retain its ability to function as a donor in the conjugal matings, right? So because donor strains with integrated F plasmid can transfer the chromosomal genes also to the recipient with high efficiency, they are called HFRs or HFR strains. Transfer of single-stranded DNA from an HFR donor to the recipient begins from the origin of transfer. Means this origin of transfer was present in the F plasmid somewhere, right? So again, the transfer will start from the ority only. But now what is the basic requirement you need to understand here? Suppose, so as I told you, this is having one origin of transfer, and origin of transfer is the sequence where your enzyme that is called relaxase enzyme will bind and it will make a complex called relaxosome, right? So suppose it is integrated. So this is F positive cell, this is HFR, because this plasmid is being integrated into the bacterial chromosome. But the things will not change. Suppose it is having ority here, and here it will bind. So this enzyme is called relaxase enzyme, and it will still make a relaxosome complex. But what will the possibility now? Once it will create a nick at this particular site, and you will see it will remain transiently attached to the 5' end of the plasmid DNA or IT DNA. And it will start transfer from 5' to 3' direction. So first of all, you will see, because this is broken from this particular site, right? So first of all, if this is 5' upper end, and this will start transferring into the some F negative cell or the recipient cell, you will find that, okay, half will transfer first, but half of the plasmid, that is the another segment of this F plasmid, will be able to transfer itself only when this complete bacterial chromosome is being transferred, right? अगर नहीं समझ में आया तो मैं दोबारा बता देता हूँ कि जब यहाँ पे निक प्रोड्यूस होगा, यहाँ पे क्लीवेज कट ये रिलैक्सेज एंजाइम क्रिएट करेगा, तो 5' एंड रेसिपिएंट सेल में पहले ट्रांसफर होगा. तो half F plasmid will be transferred first, and the half F plasmid can only be transferred only when this complete chromosome, bacterial chromosome, is transferred from the donor cell or HFR to the F negative or the recipient cell. Okay. See. So actually this is not the case. So as it is shown here, so suppose this is this arrow is actually the direction. It is moving from 5' to 3' direction, and it is bound transiently to the relaxase enzyme, right? So the half portion will be transferred first into the recipient cell, whereas half plasmid will be able to transfer only when this complete bacterial chromosome, which is shown in gray color here, is transferred, right? So we will read this now. So the transfer of single-stranded DNA from the HFR donor to the recipient begin from the origin of transfer or IT only within the F plasmid and proceed as described above, except that the transfer DNA is a hybrid replicon, which is actually made up of F plasmid, which is integrated into the bacterial chromosome, right? So this, you know. In matings, if it is between F positive and F negative bacteria, only F plasmid is transferred to the recipient. So this is the difference between F positive, F negative, and HFR F negative conjugations. If you think of mating between F positive and F negative bacteria, only F plasmid is transferred to the recipient cell, whereas in case of mating between HFR and F negative strains, the part of F plasmid is transferred last after the entire bacterial chromosome has been transferred. So this I want to tell you the half of the plasmid or the part of F plasmid is transferred at last after the entire bacterial chromosome is being transferred from the donor or the HFR to F negative or the recipient strain. Because only part of F plasmid is transferred at the start, the F negative does not become F positive, right? Unless the whole F plasmid is transferred from the donor to recipient cell. Therefore, this type cannot be regarded as that the F negative cell will become F positive after this will take place. The second problem is, so first problem you are able to understand, the F negative cell will not become F positive until and unless the second half will be able to transfer itself from the donor to the recipient cell, right? So suppose this portion is able to transfer only after 100 minutes. A complete transfer take about 100 minutes in E. coli, and the conjugation usually break before this process is finished. As I told you, there are many disturbances in the environment and the conjugal transfer is broken. This tube or the pilus is broken before the conjugation is finished. Because it is a long process, it is a time taken process, it will take about 100 minutes to completely transfer the bacterial chromosome plus F plasmid from HFR or the donor bacteria to F negative or recipient strain, right? So this is very important. This time factor is very important. And depending upon this time factor, we can perform various experiments to map the genes which are present over this bacterial chromosome, right? Thus, a complete F plasmid usually it is not feasible. The complete F plasmid is not transferred, and the recipient cell will remain F negative. Okay, so now here is a question for you. You need to understand, because this F plasmid is small in size, therefore it is most probable that if there is a meeting between F positive and F negative cell, that there are chances that the F negative cell will also become F positive. But if there is a mating or the conjugation between HFR and F negative strain, then possibility of becoming this F negative to F positive is very, very less, since it will take about 100 minutes, and this is not feasible normally in the environment. Actually, before that completion of the transfer, this conjugation tube will be broken, and therefore complete F plasmids or all the transfer genes will not be able to transfer from the donor to the recipient cell. Thus, the F negative cell will remain F negative even after a long time. Means any time you can take before 100 minutes, right? So after the replicated donor chromosome enter into the recipient cell, it may be degraded or incorporated into the F negative genome by recombination. So homologous recombination is generally the process by which these genes or DNA molecules are being integrated into the bacterial chromosome. So always there are two processes, and there is a competition between degradation and incorporation, right? So mostly the foreign DNA, if it is entering into the bacterial cell, there are enzymes, exonucleases or some other enzyme, which will degrade it. And if it is not degraded, if it is stable enough, then the second possibility is that, which is very rare though, it will be integrated by homologous recombination into the bacterial chromosome or the F negative genome. Now there is third term. So I hope up to here you are able to understand the differences between F positive, HFR, and episome, right? Now the fourth one is F dash. Now what is F dash? So suppose this F positive cell, this F plasmid is integrated into the bacterial chromosome to make an HFR. You understand this? Now it may happen that for several generations it will replicate, and then it will change its mind, and then it will come out of bacterial chromosome, right? Now this excision process is an error is an error prone process. There are mistake when this will exit out of this bacterial chromosome, and it has been seen that it will uptake some of the genes from bacterial chromosome. As you can see, one gray portion here in the black uh, plasmid or the F plasmid. Since this plasmid is not the same of this, because this was completely black and there are some extra genes which are coming from this bacterial chromosome during excision process, therefore it is given another name. It is called F dash, since it is different from the F positive or the F plasmid, right? So now the third type of conjugation which you can design as F dash F negative conjugation. And you will see again the size of this plasmid is small when compared with this HFR, and therefore there are chances that this F positive plasmid, this F dash plasmid is able to transfer itself from the donor cell, that is called F dash, to the recipient cell, that is called F negative, which is not having F plasmid. And the F dash, this F negative cell, the recipient cell, will also become F dash. So the integrated F plasmid in HFR strain can sometimes be excised from the bacterial chromosome. So it will excised, it will come out of this bacterial chromosome again. If the excision precisely reverses the integration process, so it will give the F plasmid, but this is not generally happening. So this F positive cells are produced, but on a rare occasions, sometimes. However, excision process occur by recombinations involving insertion sequences or other genes on the bacterial chromosome that are located at some distance from the original integration site, right? So they are having different genes or the sequences from where they can excise itself out of this bacterial chromosome. In such cases, a segment of the bacterial chromosome become a part of F plasmid. What will happen? Some of the genes or the section of this bacterial chromosome will become a part of F plasmid, right? Because this is an error-prone process, right? So this erroneous excised plasmid is genotypically distinct from the original F plasmid. There are genotypic differences between this F plasmid and this F dash plasmid. It is therefore called F dash. Right. So a cell containing an F dash plasmid retain all of its chromosomal genes, although some of them are on the plasmid. So actually, the bacterial cell is having all the genes, because some genes will remain here in the bacterial chromosome, some genes are remaining on F plasmid, but actually they are present since they're present in the same cell. Therefore, it is having all the genes, but when this will transfer from F dash to some recipient bacteria, from donor to recipient bacteria, it will transfer some of the genes of bacterial chromosome to that recipient, right? And they may be some resistance genes or some which are essential genes which are required for metabolism of the bacterial cell. So see here, these F dash F negative conjugation is virtually identical with the first one, that is F positive F negative conjugation, since the size is similar almost. A bacterial gene on the F dash plasmid is transferred with it and need not be incorporated into the recipient chromosome to be expressed. The recipient become F dash. So this you need to note. The recipient cell in this case of F dash F negative transfer will become F dash. It is not the F positive, right? This will become F dash because it is having the copy of this plasmid on. And it is a partial diploid merozygote, since it is having two set of genes carried by the plasmid. So there is a question in your book also. So if you find it, there is a question. A bacterial cell that is mostly haploid, it is having one set of genes, but it is diploid for some regions of the genomes, and this is called as. So there are various terms for these. This is called merodiploids. These merodiploids are also called as merozygote or partial diploid cells, right? So you need to remember this, these merozygote cells or merodiploid cells, whatever you call them, they will result because, so see here, why this is called merodiploid? Because this conjugation is actually intraspecific, means they are happening in the same species of bacteria, and same species of bacteria are having almost same set of genes. So suppose if this F dash plasmid is being transferred from this F dash to some F negative bacterial cell, what will happen? This plasmid is transferred to this F negative cell. What will happen? It will also transfer a segment of this bacterial chromosome to this cell. And this merozygotes are only formed in this F dash F negative plasmid conjugation type. Okay, because it is having some genes from the bacterial chromosome and they are also present over the same bacterial chromosome, right? Therefore, it will become partial diploid for some genes which are present over this F dash plasmid. So two things you need to remember from this discussion. One, when you do mating between F dash and F negative cell, the recipient cell will also become F dash, not the F positive, right? And the second thing, since it is carrying some genes from the bacterial chromosome, it will become partially diploid, that is called merozygote or merodiploid, whatever you call. Okay, so since it is having two set of genes carried by the plasmid, such transfer of bacterial genes is called sexduction. So this special type is called sexduction. Now, what is the application of these experiments or these gene transfers or conjugation? So conjugation can be used, especially the second one, that is called the conjugation between Hfr and F negative strains, because you need to transfer the bacterial genes from the donor to the recipient cell. And therefore, by this process, you are able to map the genes which are present over this bacterial chromosome, which are unknown. So conjugation between different HFR and F negative strains can be used to map the relative position of genes. Although these positions of genes or gene mapping can be done by using any horizontal gene transfer method, including conjugation, transduction, or trans. So I showed you this already. So we were having three methods by which the genes can be transferred between the species, between the different cells of bacteria: conjugation, transformation, and transduction. We can map the genes by all the processes in all the three ways. But here in the conjugation, we will study the gene, or we will map the genes and their position over bacterial chromosome if we do mating between HFR and F negative cells, right? So the mapping procedure is based on the fact that gene closer to the origin of replication. Okay, so those genes which are present closer to the origin of transfer, this is called ORIT, will be transferred, or they are having more chances of wait a minute. Wait a minute. Actually, my internet is fluctuating. I need to fix it. Otherwise, you will face the problem.